Check the mic to make sure. So we have so much to do there.
Is it recording now?
I'll go check that out.
I just was how long is that review session? Sorry? How long is that review session? About 50 minutes. 50 minutes?
Okay, folks, if you don't mind, just before we officially get going, I had a really quick announcement for you. So we are going to try to have an extra review session related to getting you ready for the next term test, which I believe is next Tuesday. So you have about a week. So some of you have been asking like how long this is going to be, what if you can't come down, etc. So I think I've just spoken to a couple of students who have been helping out throughout the semester in terms of recording and then uploading onto YouTube. And I think we're good to go on Friday, is that right? So we will have a streaming link for you if you can't make it down. And you can ask questions either on the YouTube link directly, so you can put your questions underneath. There'll be someone checking for that, and then you can ask your questions that way if you can't make it down. Or you can post them onto social media, and someone will be checking that feed as well. So you can follow along from home if you can't make it down. It will be recorded, and you can ask questions through those different mechanisms. Hopefully that will take care of any questions that you might have. So it should be about 50 minutes. I'm going to go through the material, the last 4 lectures, the 2 from this week, the 2 from the week before that you need to study for your upcoming term test. And the notes, again, will be available for everyone online, and there'll be hard copies for people showing up in class. Any questions before we get going? And it is over in Wetmore Hall in room 54G. Again, apologies on the time change. I had another meeting that showed up on my radar for 1 o'clock on Friday, so I had to cancel the tutorial, unfortunately. Again, apologies on that. Okay, so the next few slides are actually really important that you understand how the cerebellum is used, and there are some really important concepts that we're going to be talking about as we go through the cerebellum. So please make sure that you understand it, or that if you're not sure about it, that you circle it, come back and see me on Monday, or come and ask a question online or in person. So we talked about the cerebellum. We talked about the fact that it is not essential for motor performance. However, if you had either damage to the cerebellum where you see these white hyperintensities, and again, you don't need to worry about having an MRI, but if you see these whiter spots here in the cerebellum, this is what it normally looks like. And if you see these hyperintensities on the cerebellum, then you know that you have a cerebellar dysfunction. You can also have agenesis, where you have a loss of the cerebellum or the complete lack of the cerebellum, and this will lead to delayed motor development, and the individual will still be able to move, but will have clumsiness. Hopefully by the end of today's lecture, you will understand why clumsiness comes about. And the conductor and the cerebellum is viewed more as the conductor of the system. It doesn't generate any movements by itself, so it is not a movement generator. And by the way, for your term test, you should make sure that you know what sorts of things in the brainstem, midbrain, motor cortex are involved in generating different types of movements, things like the mesencephalic locomotory region and other types of nuclei. But the cerebellum is not one of those. Okay, and the cerebellum is there just to help with the coordination of different types of circuits. The cerebellum is different than the cerebral cortex. It still is very similar in the sense that there are two different sections to this. So if we were looking at the cerebellum in cross section, and you'd have to imagine that up here at the top, we would have the cerebral cortex. But if you were taking a look, if you took a sagittal section, a sideways section through the cerebellum, and this was the brainstem, and this would represent the pontine nuclei, and this is the cerebellum in cross section, we would see that there are two structures that are very similar but not identical to what we would have in the cerebral cortex. In the cerebellum, we have this cerebellar cortex, and we will look at the individual circuits that are involved in the cerebellar cortex. And we would also see that we have these cerebellar nuclei. Similar to what the cortex looks like in the cerebrum, we would also see that we have lots of foliation. It looks like it has these sulci and gyri as well, but the structure is very, very different, and we call these folia. It looks more leaf-like in appearance than it does in the cerebral cortex. So the most important parts are these cerebellar cortical neurons that we're going to be looking at, and then also the cerebellar nuclei that we're going to be looking at as well. They are very different, and you need to understand different things about these different parts of the cerebellum. The cortex, when you're studying it, you need to understand the circuitry that's involved. The nuclei, where we have a set of 4 different nuclei that we'll talk about in a moment, you need to understand what their major functions are as well as their major characteristics. Okay, so keep that in mind as we're going through the next few slides in terms of the major points from this. The other thing to take away, because it segues really nicely into the next slide, is that we have a lot of inputs. We have a lot of sensory inputs, proprioceptive inputs that are coming in to the cerebellum from multiple sources. So the vestibular nuclei about balance, it's sending projections into the cerebellum. We will also see other sensory information coming into the cerebellum as well. So there are lots of things to consider, and in the next slide we're actually going to be looking at what sorts of sensory input come in to the cerebellum. So what kind of information is being integrated at the level of the cerebellar cortex, in other words? So we have a number of different regions, and just to orient you a little bit, instead of looking at it from the side now, we are looking at the cerebellum face on. So if someone were standing right behind me and then taking a look at my cerebellum, this is what they would be able to see in terms of the cerebellar structure. So we have in the midline, right in the middle, we have this structure known as the vermis, and going more laterally from the vermis, we will have these different types of lobes. We'll have an anterior lobe, and then we'll also have a posterior lobe associated with this as well. The most important thing from this particular diagram, though, that segues from that previous slide, is that different types of input, sensory, proprioceptive, all sorts of sensory information that's coming in are going to different regions. So, for example, if we were to take a look at the vermis, it receives a lot of vestibular information, things about your sense of balance, right? Remember the vestibular apparatus, it's all about the sense of balance, and you can see that the vermis is receiving information from the vestibular system, including the vestibular nuclei. It is also receiving auditory input as well as visual input. And you can see auditory visual here in these lines. And you can see that, again, this is not in the same areas. It's widely distributed. There are somatosensory inputs coming in from the spinocerebellar pathways that are coming in. So the cerebellar cortex is receiving lots of information about where your arms are in space, how you're standing upright, your sense of balance. It's receiving information from the spinocerebellar pathways that we didn't go into in a lot of detail. But all of this is being integrated at the level that you see here. And the big structure, even though it's not the only area that's receiving it, the big structure that seems to have a lot of this input is this vermis layer that you see here. So it has a little bit of somatosensory, it has vestibular, it also has a little bit of auditory visual. So we have a lot of input coming into the cerebellum, but a lot of it is going in the midline at the level of the vermis. Okay, so those two slides, the one that I showed you previously and this slide, kind of go together. And what I want to do now is, when that information is coming in, what that information looks like. So I'm going to come back to this cerebellar nucleus later. I told you that there is a circuit that you need to worry about, and we're going to be looking at the cerebellar cortex, which is shown up here at the top. And then we're going to be looking at these cerebellar nuclei separately in a few slides. So for the time being, you need to understand these circuits that are bringing this information in. So the first thing to consider is that you saw a lot of inputs coming into the vermis, and both in that last slide and the slide that came before it. That information that is coming in from the spinocerebellar tract, from the vestibular nucleus, the somatosensory areas, the proprioception, all of that is coming in to the cortex through what's known as the mossy fibers. These mossy fibers are those axons that we saw in this diagram here. These would all be mossy fibers. These mossy fibers are making particular and specific inputs to the vermis here. And if we take a look at it as a circuit, we would see these mossy fibers coming in, sending their axons in, and then they would make synaptic connections onto the next cells within the cerebellar cortex, known as these granule cells. And this is the granule layer where they are making their first inputs. So proprioceptive inputs are coming in, somatosensory inputs are coming in, auditory, visual inputs, they're all making their synapses here at the granule layer, and all of those axons we are calling mossy fibers. Do you have any questions about this? So sensory input is coming in through these mossy fibers. A really quick one, yeah. So we're starting at the bottommost layer. So I'll get to those layers in a few moments, okay? So this granule layer is the first layer that we need to be concerned with. And then from the granule cells that you see down here in this layer, we're also going to see another set of synapses that go up. And you can see that the axons of the granule layer are the next sort of synapses that we're going to be taking a look at. And they send up their axons, and they produce what are known as these parallel fibers on the upper cerebral cortical layers. So these are all the axons from this one particular granule cell. And this is another one that's going up, and they form their own axons, which are known anatomically as these parallel fibers. So mossy fibers are different. Parallel fibers are different. You should know which cells or what sorts of information or where they're going in the cerebellum. Okay, so these mossy fibers that we saw over here are all those inputs from the spinal cord, brainstem, cerebral cortex, motor signals, etc. And now we're going to be looking at this next set that are sending up these axons up here. So this would be the second layer, and then we'll see how they make synaptic connections onto the output cells of the cerebral cortex, which are the Purkinje fibers. So far we've had mossy fibers, then they're making connections onto these granule cells. These granule cells then send up these parallel fibers, which make the next set of connections onto these cells known as the Purkinje cells. And these Purkinje cells have this really large, elaborate sort of synaptic tree associated with it. And you can see that many of these parallel fibers will make synaptic connections onto these Purkinje cells. So why are these Purkinje cells so important? They're going to be integrating all of that sensory information from many different different granule cells. And in addition, these are also the output neurons of the cerebellar cortex. They are sending a signal outwards now. Okay? They're not just having signals coming in. These are the ones that are going to be sending signals out. So a couple of really important things about these Purkinje cells that we're seeing here. So here's one Purkinje cell, here's another Purkinje cell, and here is yet another. These Purkinje cells are in this middle layer. They're receiving all of these different inputs from these parallel fibers, which in turn received all this information from these mossy fibers. But what they're doing is actually they are sending out inhibitory axons. So you can see they are sending out an axon here. They are going to release GABA, which is an inhibitory neurotransmitter, and their major function is to inhibit these different cerebellar nuclei. And these cerebellar nuclei that I'm going to get into on the next slide have very, very specific functions. Remember, if your cerebellum isn't working, you can move perfectly fine, but you're going to be really, really clumsy. You're not going to be able to grasp for things easily. You're not going to be able to swing a baseball bat really smoothly, and all of your motor functions are going to be just slightly impaired, and you're going to be a little bit clumsy. So these are going to be integrating all of this information that has been coming up and then sending information outward, and they are going to be inhibiting through the release of the neurotransmitter GABA these different cerebellar nuclei. In turn, these cerebellar nuclei also send projections to these other motor centers that are involved in motor activity. So why are these output neurons so important? So there's a couple of really key things. I told you at the outset, make sure that you understand this circuit and what this circuit does, how it receives input from these mossy fibers, that there are parallel fibers, that the output neurons are the Purkinje cells. These Purkinje cells will then inhibit or turn off specific cerebellar nuclei. And now I told you also to know the characteristic features of these cerebellar nuclei. So why do we want to be turning off these cerebellar nuclei? The neurons that are found in these nuclei are spontaneously active. They're tonically turned on all the time. Their job is to excite different motor centers. So in other words, they're always turned on when you're moving. When you want a motor program turned on, the cerebellar nuclei that you see here are the ones that are continuously active. In order for you to turn off a motor activity, in other words, when you're reaching for something and you want to stop precisely, you need your cerebellar Purkinje cells to turn off specific groups of these different cerebellar nuclei. And they're all very, very specific. So for example, you might find these fastigial groups over here, the fastigial cerebellar nuclei, which are always active, they're tonically excited, they will turn on motor programs that are related to posture as well as locomotion. So if you want to stop your locomotion in a very precise way and turn off that locomotor program, you need the fastigial nuclei to be inhibited through the release of GABA from the correct sort of groups of Purkinje cells near the vermis layer. If you're looking at the interposed nuclei, which are consist of both globose as well as the embelliform cerebellar nuclei that you see here, and these are together called the interposed, and you can separate them out into the embelliform and the globose. Oftentimes we see these receiving inputs from the intermediate part of the cerebellar cortex, and when these cerebellar cortical Purkinje cells turn off these embelliform or globose groups of cerebellar nuclei, one of the things that they are important for is both reaching for something as well as grasping something. And in other words, they are involved in your upper motor neurons to reach for and grasp something. If you don't turn them off, you may overreach for a particular object. So I'm trying to reach this particular microphone, and if I don't turn it off at the precise moment, in order to grasp it, and I don't turn off the globose and embelliform, I will overreach it, I won't have stopped in time, and I won't be able to grasp it accordingly. Hopefully now you understand why the cerebellum is important for clumsiness. If it's not working right, you're going to be overreaching, not grasping at the right moment, etc. On the most lateral part, the Purkinje cells will inhibit the dentate nucleus, which is important for very, very fine motor skills. Things like writing and being able to write precisely, your speech and being able to pronounce words correctly and be able to speak correctly are all due to inhibiting at precise moments these dentate groups of cerebellar nuclei. So if you put all of this in together, what this means is that a cluster of these Purkinje cells will be receiving all this input at any given moment, that I'm reaching for something that I need to be able to turn off at a specific moment in time. And so it's controlling a specific motor performance. So reaching for something, saying something, or being able to write something is a group of motor activities that I need to be able to regulate. These Purkinje cells, in other words, are taking in all that information about what my muscles are doing while I'm reaching for something, or how I'm walking. All of that information is being targeted to these Purkinje cells, which in turn will turn off specific cerebellar nuclei at just the right moment in order to give you those really precise types of actions that allow you to end these at very, very specific times. So you can imagine, for example, playing the piano. There are certain groups of nuclei that will be involved in being able to turn off your motor programs at precisely the right time. And again, you'll need to understand the nuclei that are involved, why turning them off at the right time is important for specific motor activities, and how that circuit plays a role in all of this. So again, not to spend a lot of time on this, and I'm not going to be asking you for definitions. I think that if you get something that is an application, it's a good idea to make sure that you are familiar with these different terms. But overall, these are all related, where you're either not having a particular type of movement or having a movement kind of go back and forth or meander because you're not having coordination, overshooting a target, etc. All of these are due to the fact that the cerebellar circuit is not working, or you're not able to turn off these nuclei at specific moments. So do you have any questions related to this section at all, about the circuits, which you definitely have to know, or about the functions of these nuclei that are being turned off? A really quick question. That's correct. Yeah, so there's a topology, in other words. The lateral parts will inhibit specific nuclei. The vermis part will inhibit other groups of nuclei, and then the embelliform and globose will be inhibited by others. Any other questions? Okay, so I'm going to move through a couple of things just to give you an overview of different sort of aspects of the autonomic nervous system. And I actually don't want you to be thinking about it sort of as a separate nervous system, even though it's presented to you, and I'm going to lecture the next section as a sort of separate type of nervous system. In fact, there's a lot of similarities between what we've already talked about in the somatic nervous system. So when you think about a reflex, you're actually not changing or using a different group of muscles to cause your arm to kind of jerk back or to hold up a load. It's the same muscles that you use all the time. And the autonomic nervous system, again, we can use it for a reflex activity, or we can understand it from the basic circuit of what the reflex would look like. So I'm going to go through some of the general features of this system, different examples of autonomic nervous system reflexes, and then go at a higher level to talk about different autonomic nervous centers within the brain, and then talk about some of the physiological consequences when these autonomic circuits are not necessarily working well. So I'm going to give you a really quick overview of the different types of the autonomic nervous system. A lot of you will equate the autonomic nervous system with the fight or flight, the stress response. When you're thinking about autonomic nervous system, you're talking about blood pressure increasing, you're talking about getting your pupil as wide as it possibly can, you're thinking about increasing your breathing rate. But the reality is, if you did not have the other part of the autonomic nervous system, the parasympathetic nervous system, you wouldn't be able to survive for very long periods of time. And this is because this fight or flight is designed to be a short-term solution to helping you overcome a crisis. But the reality is, if you don't replenish all the energy that you're expending in a fight or flight through the parasympathetic, the housekeeping role of the parasympathetic, then you're not going to be able to do the fight or flight for very long. So again, you don't have to worry about memorizing this. This is really just to give you an overview that this autonomic nervous system, the moment you start exercising, the autonomic nervous system kicks in almost like a reflex so that your blood is automatically diverting to the muscles that require more blood flow. You're going to be stimulating sweat glands, all the function of the autonomic nervous system. So emotional responses will also turn on the autonomic nervous system, right? So you don't have to just be exercising to get increased blood flow. If you think of something really scary or something that is really stressful, you can also increase your blood flow, increase your heart rate, irrespective of having to exercise. And again, changes in body position. Every time you sit up, you regulate your blood pressure. It's done automatically as a reflex. Eating, it may not seem like an autonomic function, but I'm going to suggest to you that it is an autonomic function, that it's controlled by a group of neurons, and that when you have problems with anorexia or hyperphagia, where you're overeating, or if you have other types of issues with the signaling regulation, that all of this eating itself is also a function of the autonomic nervous system. So again, you don't have to worry about this diagram. It's just to give you an idea of what sorts of things we're going to be taking a look at. So up until now, we've been looking at what's known as the somatic nervous system. So we've been looking at how motor neurons become activated. We've been looking at the sensory pathways that send information up through the thalamus, the cerebral cortex, and then all the pathways that come back down that allow you to do something with it, how you do reflexes, how you decide that you're actually going to engage a motor program. And that's been the last 2 lectures that we've been looking at. And we've really neglected this side of things. So we need to go back and re-examine how the autonomic nervous system is actually in many ways related to the somatic nervous system that we've been looking at so far. And there are lots and lots of parallels that we can draw along the way. In fact, there are reflexes in the autonomic nervous system, just like there are reflexes in the somatic nervous system. When you have the muscle spindle stretch, or you have the Golgi tendon, we have very similar types of reflexes in the autonomic nervous system as well. And we'll see that there are higher order processes from the hypothalamus and the reticular formation in the brainstem that will also influence in a descending way, similar to the motor cortex, etc., the autonomic nervous system that we're going to be taking a look at. So again, this is really just overview for you, so don't spend any time at all kind of memorizing this. I'll tell you when you should start thinking about what you need to know about the autonomic nervous system. This is the stuff that you use when you're controlling your internal organs, when you hear about the endocrine system in the next group of lectures, when you're looking at changes in blood flow. We'll look very briefly at how the autonomic nervous system controls smooth muscles in various parts of the body, including the viscera, your gut, as well as in the eye. And all of this is there to control the internal homeostasis of your body. So we're going to see that there are 2 different efferent outflow going out from the spinal cord outward to these different structures that I've just mentioned to you, 2 efferent systems that work in parallel. They are not antagonistic to each other. They are actually working in a complementary fashion. And we'll see a sympathetic autonomic nervous system branch, and then we will see a parasympathetic autonomic nervous system branch, and they are related. You need to have the parasympathetic to allow the sympathetic to work. Again, sympathetic being more stress related. People used to think that it does all the hard work and that the parasympathetic was there just for housekeeping, but it turns out that this restorative process, after you've just run a 26-mile marathon, you need to be able to recover. If you just kept running marathon after marathon without any time to recover whatsoever, your body wouldn't last very long. And it's the same thing. When you have this consistent, continual fight or flight, where your sympathetic is on all the time, unless you have a way to recover and recuperate from this, you're not going to be lasting for very long periods of time. And again, this is one of the reasons, if you're wondering, why, for example, the University of Toronto and most universities are actually spending so much time trying to figure out ways to help you as students to manage your stress levels, to make sure that your fight or flight system isn't on all the time, especially here at U of T, where stress levels tend to be a little bit higher on average, especially in the life sciences, when you compare it to the rest of the different universities or the student population as a whole. So again, this gets into this fight or flight. You'll hear about this rest and digest later on in the course. And these are sort of the two different branches. Now we're going to be spending some time looking at how this all works. And again, I'm going to come back to this sort of topography several different times. For the time being, if you want to kind of jot this down, this is something that I do expect you to be able to tell me a little bit about. When we are looking at the autonomic nervous system, we are always, depending on what we're looking at, whether it's sympathetic or parasympathetic, it makes no difference. In the autonomic nervous system, we always have a preganglionic fiber, which comes from the central nervous system, generally the spinal cord, but not always, and you'll understand why I've said that in a moment. You always have a preganglionic fiber, and you always have a postganglionic fiber. Again, same thing for sympathetic, same thing for parasympathetic. Both systems, you have pre and postganglionic. At this preganglionic synapse that you see here, it is always going to be releasing acetylcholine. It doesn't matter, again, if it's sympathetic or parasympathetic. It always releases acetylcholine. So preganglionic fibers within the autonomic nervous system always release acetylcholine. You guys have learned previously with Dr. Key about different cholinergic receptors. So which receptors that bind to acetylcholine are you guys aware of? So there is the nicotinic, I think I've heard that response. And what's the other one that binds to acetylcholine? So it's the muscarinic. So think a little bit about this. You have choices of two different types of acetylcholinergic receptors. So acetylcholine gets released here, it has to activate this neuron, and you have receptors for acetylcholine on this postganglionic neuron. What kind of receptors do you think we find here? Again, you have a choice of two, either muscarinic or nicotinic. So what does nicotinic do? So acetylcholine binds, it binds to the nicotinic acetylcholine receptor, then what happens? Does it activate a G protein or does it activate a channel? So it activates a channel, it opens up a sodium channel, and it causes sodium influx. The muscarinic type actually causes G proteins to become activated, and depending on what happens downstream, it can either inhibit or excite. So on these postganglionic neurons in the autonomic nervous system, we always have the nicotinic receptor that is found here on the postganglionic cell. Okay, and it's very important that you keep that straight. We need fast responses, we need nicotine, we need ligand-gated ion channels to be opening up. Any questions on that before I get going? Okay, so it's always acetylcholine between the pre and post, always binds to a nicotinic, but what happens after that is a little different between the para and the sympathetic nervous systems. So any questions about that before I get going? It's a really important concept. I want you as physiology students, when you leave the course, you should remember that sort of topography of the responses. So this is the generalized feature of the autonomic nervous system. So you'll see the sympathetic nervous system over here on the left, and you will see the parasympathetic nervous system there on the right. If, again, before I get into more details, we always see coming out of the spinal cord with the sympathetic, we always see preganglionic fibers. They are making synapses on these little round balls that you see here, and I'll get into that in a moment. And then we also have from the parasympathetic, we also have preganglionic fibers, but they're not doing exactly the same thing. We still have pre and post, okay? Is there anything that you notice about where these axons are coming from the sympathetic versus the parasympathetic on the right? Any differences that you can see right away? because they're targeting the same organs. We're looking at liver being innervated by both the sympathetic and the parasympathetic. The trachea is innervated by both the sympathetic and the parasympathetic branches. Anything that you see different between the left and the right-hand side? No overlap, okay? True? True dat, no dat. Absolutely. What else? So if we were to take a look at this, there are some key features that, again, Max had pointed out. There's no overlap, but there's a couple of other things to make note of as well. So if we're to take a look at what we're looking at, one of the big features that you should observe is that in the sympathetic nervous system, all of the neurons that are coming out of the spinal cord are all at one particular level. They're either at the thoracic level or at the lumbar level. They all come out of basically the same place. They may go up and innervate things like the eye, up in the head, but they're all coming out of the thorax and the lumbar region. So we say that their origin, the preganglionic fibers, are all coming from the thoracolumbar region of the spinal cord. Again, if you were to take a look at where the origin of the parasympathetic nervous system is, they're coming out of the brainstem as well as part of the spinal cord, which is known as the sacral region. So in your textbook, you might see this being represented as the craniosacral origin of the parasympathetic neurons in the parasympathetic system. Okay, so on the left, in the sympathetic, we have the thoracolumbar origin. They only come out of that region of the CNS. And for the parasympathetic, we say that it comes from the cranial sacral region. So the brainstem as well as the sacral region of the spinal cord. And that again is something that you'll see again in your notes. It's an important sort of feature and defining feature of the autonomic nervous system. So if you take a look, there are some other key points that you will see here. The solid lines, whether they're the solid lines that you see in the sympathetic or the solid lines that you see here in the parasympathetic, the solid lines represent myelinated preganglionic fibers. The preganglionic fibers are always myelinated. Okay, so that's another feature that makes them unique. The postganglionic fibers that you see are always unmyelinated, and they're represented in these dashed lines. Is that clear for everyone? And is it something that you can see directly on this particular slide? So I also want you to think a little bit about this, because this is the last point here. There are some differences, though, in the lengths of these pre- and post-ganglionic axons. So these are the preganglionic axons, makes a synapse right here, another preganglionic, another one right here. Are the preganglionic axons in the sympathetic nervous system long or short? So they're relatively short, myelinated but short in this sympathetic nervous system. Take a look at the parasympathetic preganglionic axons. These are all of the preganglionic axons that are coming out. We know that they're myelinated as well. Are they relatively long or short? So comparatively, these are much, much longer preganglionic axons that are myelinated. The postganglionic axons are the exact reverse. So the postganglionic axons in the sympathetic nervous system tend to be relatively long. They have like long unmyelinated fibers to eventually make synapses to their various targets. Things like the trachea, the heart, the liver, the stomach, etc. All of them tend to be fairly long when we take a look at them. But the parasympathetic, if you look at the dashed lines representing the postganglionic fibers, and here is the postganglionic fiber going to the eye, to the lacrimal glands, salivary glands, etc., or to the heart, they tend to be very, very short. So there are some fundamental structural differences between the parasympathetic and sympathetic. All innervating the same tissues within your body, all of the visceral organs. Any questions on this before we go on? Or anything that you are unclear about? Again, just to make sure that you're all okay with this, if I were to take a look at this preganglionic fiber here, what would the neurotransmitter being released here be? So it's acetylcholine. And if I took a look at the parasympathetic fibers over here, and I looked at the pre and post ganglionic connection, what would the neurotransmitter being released here be? Also acetylcholine. And what is the receptor that it's acting on? Always nicotinic. Okay, perfect. So this again is diagrammatically showing you pretty much the same type of thing, that these parasympathetic fibers that we see here, it's always going to be releasing acetylcholine. So if it's not clear for you, acetylcholine is being released here. You can see that it is a long, relatively long myelinated fiber, and then it makes a connection to a short postganglionic parasympathetic axon, which is usually unmyelinated, which is what you're seeing in gray. The sympathetic is different. Do you have a question at the back? No? Okay. The sympathetic is a little bit different, where the myelinated fibers are relatively short, also releasing acetylcholine here, and then the postganglionic axons tend to be longer, unmyelinated but longer. So again, diagrammatically showing you exactly the same thing. Now, if you're looking really carefully, and this is where the autonomic nervous system gets a little bit more complicated, depending on the type of tissue that we're looking at, the next group of neurotransmitters that's being released is going to be different. In the autonomic nervous system, we are going to be releasing acetylcholine again. And in the sympathetic nervous system, we're always going to be releasing norepinephrine or epinephrine. Okay? So these neurotransmitters released from the postganglionic neurons are different. Any questions on this before we go on, or anything that I can clear up for you before we get into some more specific details of the sympathetic versus parasympathetic? Okay, so again, this is just a different way to show you exactly the same thing, so you can see that it's a really important concept that I want you to make sure that you are aware of. We've been spending time previously about this somatic motor system in the spinal cord and how it causes contraction of skeletal muscle. And we looked at some of the pathways there. And what we're looking at now is something very similar. And we're going to take a look at this in some more detail about how specific neurons in the CNS, and in this particular case, we're likely looking at the sympathetic nervous system, how they send out projections, and they will not work on skeletal muscle. They'll be working on all sorts of different cell types, whether it's smooth muscle, whether it's cardiac muscle, whether it's working on fat cells. The targets are different, but the concept, the idea, the overall way in which it's all put together is pretty much identical. And we'll also see some regulation that later on in today's lecture coming up from higher levels of the CNS. Again, you don't have to worry about trying to memorize everything on this diagram. I'm not going to take anything specifically from this diagram that I haven't covered on your next term test. So what I'm going to do now is go over the sympathetic nervous system, revisit some of its key features, talk to you about some of the specific aspects of it, and then move on to the parasympathetic as well. So the sympathetic nervous system is actually kind of unique. So the sympathetic nervous system, when you activate it, when you get a stress response, generally speaking, it is a generalized response, which means that everything turns on. You can't just turn on like one system. You can't just have your pupil dilating and that's it. If your sympathetic nervous system is on, your respiration is going to go up, you are going to be sweating, you are going to be increasing your heart rate, blood pressure is going up, pupils dilate, everything is turned on at the same time. It is a generalized response. The parasympathetic is not that case. You can turn on different segments of the targets. You don't have to turn everything on. So when it's generalized, it means everything turns on. It's not a segmented, localized response that the parasympathetic is going to have. And this includes things like increased alertness. When you are activating the sympathetic nervous system, its job is to increase alertness to make sure that there's as much light as possibly can come into your eye as possible by increasing the pupil diameter. It's going to increase energy reserves and release energy reserves from your adipocytes, from your liver, etc. It's going to make you feel good temporarily. It increases a sense of euphoria. It will increase your cardiovascular system, the respiratory activity, more breathing, higher heart rate. All of this, again, is related to sympathetic nervous system activity, elevation in muscle tone, and mobilization of energy reserves. I don't know about you, if you've ever gone to the exam center, you once in a while sit beside one of your colleagues who has this like during an exam. And you know that they might have like this elevation in muscle tone and everything else. If you ever sat beside someone who's like really pounding away with their foot while they're writing, and it's like a little bit distracting for you, and this is sort of the sympathetic nervous system where the muscle tone is increased and the motor activity is increasing as a consequence of them being nervous and then activating this sympathetic system. Sorry? Are we good? Okay, perfect. So a couple of things to make note of. This sympathetic nervous system, when we look at the efferents, the preganglionic fibers that come out, these preganglionic fibers, the preganglionic neurons are efferent neurons. They send signals away from the nervous system, therefore they are efferents. And these efferents all arise from a region of the thoracic lumbar cord, the thoracolumbar spinal cord, which is found in the intermediolateral portions. And I'm going to show you this on the next slide, so you should link this slide with the one that comes after. And then they will send out very short projections, myelinated axons, to a chain of neurons that are very nearby the spinal cord that will then send their projections. So these are the postganglionic neurons that we're talking about in this synapse, the chain of synapses beside it. And then these postganglionic efferents from these different ganglia will then target different types of tissue. It's not always another nerve. It could be, for example, fat cells. It could be smooth muscle cells. It could be a number of things, right? So we'll talk about that in some more details, or it could be a gland. This is the efferent that I just mentioned to you in that previous diagram. This is that intermediate lateral horn region that I'm talking about. It's found in the thoracic region as well as in the lumbar region, therefore thoracolumbar. So it's in the intermediate and it's lateral or to the side. So if it was this way, it would be medial. This way, it is lateral, and you can see where it is beginning. So it's not in the dorsal horn, nor is it in the ventral horn. It's in this intermediate or middle region off to the side. So it is sending out its projection this way. We know that this is myelinated or unmyelinated? So this is myelinated. It doesn't go very far outside of the spinal cord. It goes only a short distance. It is going to be releasing acetylcholine like every autonomic nervous system neuron does, and then it will act with nicotinic receptors, and then the sympathetic ganglion, whatever its target is, it will be releasing noradrenaline onto its target. So again, during early development, sometime between day 40 to about day 60 in development, after the neural tube has formed, your spinal cord has formed, and as you get the migration of the neural crest cells away from the neural tube, they will sit right beside certain segments of the spinal cord, which is why we have this sort of structure that you see here. You can actually see the ganglia in this particular photograph where they're very, very close to the actual spinal cord. So the spinal cord would be off here, and you can see these different ganglia. And I'll leave that for you to take a look at later. It's that short distance. And again, the sympathetic nervous system, the major influence of activation of this sympathetic nervous system is to cause the release of noradrenaline. And if we're releasing noradrenaline onto muscle and onto blood vessels, it will cause vasoconstriction. The blood vessels will be more constricted. And what this does is it will activate blood pressure. So the smaller the diameter of your blood vessel is, the more the pressure inside that blood vessel is. And we know the sympathetic nervous system, one of its functions is to maintain blood pressure. and this noradrenaline will help to do this. And it is incredibly important. Your sympathetic nervous system is not just when you're doing fight or flight. Right now, as you're sitting here, where you're not, hopefully, not too stressed out, not exercising or doing anything else, your sympathetic nervous system is there maintaining a certain tone or blood pressure that you all have. And if that were to completely disappear, all of you would be falling over fainting. And it's this basic vasomotor activity, or the baroreflex activity, that allows your blood pressure to be maintained at high enough levels so that you are perfusing your tissue, including your brain, with blood. And I think it's actually a good time to take a break. So we'll come back and we'll go through some other segments of the sympathetic system. Okay, if you don't mind, I'm going to just go through this summary diagram. It's a good summary diagram for you if you want to summarize all the things that we've just been talking about related to the sympathetic nervous system. This is a really good diagram for you to have that really covers everything that we've just mentioned over the last maybe 10 or 15 slides. So we're looking at this sympathetic division of the nervous system. This is its origin. The only other thing that you would want to put down here is what part of the spinal cord this comes out of. And in the thoraco, which is T1, lumbar, which is what the L represents, in the thoracolumbar region, which you can't see very well here, but sort of the upper part of your spinal cord around the chest, all the way down to the small of your back, the thoracolumbar region, from the intermediolateral portion of the spinal cord in this area, the preganglionic fibers will then send out very short myelinated fibers to the nearby sympathetic chain. We won't worry about the collateral chain, or they could be targeting specific glands like the adrenal medulla. And you'll hear more about the adrenal medulla in the next section when you hear about endocrinology. The neurons in this next section will then send out their fibers, unmyelinated fibers, to their target organs, whatever their targets are, whether it's in the thoracic cavity, whether it's up in the head, whether it's inside your abdominal cavity or to different limb regions, etc. Whatever it is, these postganglionic fibers will send out their unmyelinated axons, and they will release noradrenaline, or adrenaline, if you want to think of it that way, to their targets. And this is really what I wanted to talk to you about, to make sure that you are aware that, for example, the adrenal medulla, which is also part of this system in the sympathetic nervous system fight or flight, that it also uses the same system. It's also causing the release. Once it's activated, there are no more postganglionic fibers, but it's going to release noradrenaline and activate the stress response in exactly the same way. So that's why we consider it as part of this sympathetic nervous system response. And again, this is sort of a really rapid overview of what happens in the sympathetic nervous response. So the parasympathetic, we've already covered most of the major features, but if we're to take a look at this, just again, hopefully as a reinforcing review for you, where these preganglionic axons are coming out of, it is at the level of the brainstem, so we call these cranial, or at the sacral cord, and it's also coming out at the sacral cord in the intermediolateral regions. Just like we saw with the thoracolumbar, it's also coming out of the intermediolateral part in the spinal cord. But in the parasympathetic, we also have things coming out at the level of the brainstem or the medulla. And again, these are the motor nuclei that they all originate from. So they will travel longer distances, longer myelinated axons will travel to the ganglia that are actually usually embedded in the target organ. So the target organ might be the SA or sinoatrial node within the heart to increase heart rate or decrease heart rate in this particular case, as well as the enteric nervous system within the gut. And in all cases here, these postganglionic axons are going to be releasing acetylcholine. So sympathetic, the postganglionic releases noradrenaline. So in the parasympathetic, it is releasing acetylcholine at the postganglionic to its target tissues. Whether the target tissue is the heart, whether it's another target organ, it's always acetylcholine. So in other words, this system uses acetylcholine and acetylcholine. The sympathetic uses acetylcholine and noradrenaline. Any questions about this before I go on, or anything I can clear up for you in this section? Okay, so again, in terms of the functional overview, and you don't have to worry about trying to memorize this. Hopefully, it's all common sense. Um, When we're looking at the sympathetic effects, we're looking at how we increase cardiac output, which is heart rate and the stroke volume, how vigorously the heart actually is pumping. We want to vasoconstrict in the skin. Going to be sweating more as a result of this activation. And the parasympathetic is where digestion is being promoted. You're going to slow your heart rate down. It's where you're going to feel like you're in a more relaxed mood. You're going to be slowing your breathing as well. So there are a number of different reflexes that we're going to be taking a look at that revolve around this system. And just like we looked at with the reflexes in your skeletal muscles, it's the same idea that we use this circuit to generate an automatic response that your nervous system doesn't have to worry about too much, and it's done in a local circuit. One of these associated circuits is this enteric nervous system. And if you're still in the course, by the time you get to PSL301 and you're doing the digestive system, you'll learn about how the regulation of the digestive system and the visceral muscles takes place through the autonomic nervous system working on this enteric nervous system. Your gut is actually known as the second brain. You have almost as many neurons there. Outside of the brain, this is like the biggest sort of area where we have nerves clustered around. Every single neurotransmitter that you have up in your brain, you also have in your gut, every last one. In fact, we found new neurotransmitters at the gut that we eventually found also existing in the brain. The enteric nervous system is actually a very complex system. The parasympathetic nervous system will segment onto the nervous system that is embedded within the gut wall, which is the enteric nervous system. When you hear about enteric, it means inside the gut wall. This nervous system is regulated by the parasympathetic, which will speed it up or slow things down. And this parasympathetic system generally increases activity of the enteric to activate cholinergic motor neurons, and the sympathetic actually turns this off. When you're running away, the last thing that you need to be doing is digesting food. You should be like trying to get away as fast as you possibly can, and then when you're able to recover, you should be digesting as much as possible. And this enteric nervous system, again, is there to program what is known as peristalsis, the movement of your gut muscles, as well as segmentation, which is another sort of movement that your gut makes. I'm going to be really honest with you. I'm not going to be testing it all on the term test, nor about this on your final exam. It's really just to give you a heads up when you eventually get to this system, that this is the sort of regulation by the sympathetic and parasympathetic along these particular routes. And it's just to make sure that you are aware of this. So I'm going to talk to you a little bit now about the autonomic nervous system. So we talked about sympathetic fibers, parasympathetic. Yep, do you have a question? I have no idea why my live stream screen is blank. Okay, I have no idea. No idea. Do you guys have any ideas? So apparently the live stream is not live. You know, right now you should have this little thing going up there. We're experiencing technical difficulties. Can you hear things on the live stream? No idea. So if you're watching, we're doing lots of really fun things, and we had lots of really cool images. Too bad you missed it. So if we're talking about these autonomic reflexes, they work in the same way. They use the sympathetic and parasympathetic. They just happen to use it sort of in a reflex arc, just like we've seen previously. Zaid, do you have any ideas? I know as you were watching him kind of jumping down, you were thinking about all the physiology that was involved, like the cerebellum, the motor planning, the fact that he had to actually engage all of these premotor programs in order for him to do that. So pretty remarkable. The reflexes, the extensor thrust reflex that you saw him when he landed, right? I know that goes through your mind all the time. So, how about you check in about 30 seconds? I'm just going to go on, and hopefully it'll work. Yeah, you have to give it about 30 seconds. Okay. All right. Okay, so we're going to talk about these autonomic reflexes. We've already seen one of these autonomic reflexes. And the autonomic reflex that you're seeing there is the dilation of the pupil. This actually makes use of the fact, and this dilation of the pupil that you see in this individual here on the left, and the normal sort of pupillary reflex response when you shine light in. We've looked at that previously. When you shine light into someone's eye, that you should be seeing this pupillary reflex that you see here. Any ideas what's going on here in the left? Sorry, he's no idea. Someone must have an idea. Yes. Okay, sure. What kind of overdose? Okay, could be MDMA, number of other things. It could be where you have too much noradrenaline around. So oftentimes if you're going in and you're getting your eyes checked, they'll induce this type of response where they dilate your pupil as much as possible to see at the back of your eye. Again, I tend to talk a lot about physiology wherever I'm at, and one of the things that my brother does is he's a cop up in York region, and he pulls over different individuals, and he can actually tell based on like looking into their eyes whether or not like they have a fully dilated pupil where it looks really unusual that they might be on different types of drugs like cocaine, etc. So this causes what's known as a chronic pupillary dilation. It makes use of the fact that we have these neurotransmitters, and these neurotransmitters that are released by the sympathetic and parasympathetic nervous systems will actually elicit the same sort of reflex activity. And we've gone through this particular reflex before in a slightly different way earlier on in the course when we looked at vision. A couple of things I'm going to ask you to make note of, and I told you at the time that we would come back and talk about this. If you were to go back and look at your notes related to the retinal ganglion cells, we mentioned in passing that there was a small group of these retinal ganglion cells, about 20% of them, that actually contain a pigment known as melanopsin. And this melanopsin containing group of ganglion cells that sit there in your eye are actually there to detect these general light levels. And we are going to come back and talk about them because these ganglion cells are actually really important for autonomic functions, including the synchronization of your circadian rhythm with daylight around you. Without these melanopsin containing ganglion cells in your eye, you wouldn't know when you should be going to sleep nor when you should be waking up. When it's like light out, when it's dark out, you wouldn't have that synchronization. So these melanopsin containing ganglion cells, the 20%-ish that we talked about earlier, are really important when we talk about this synchronization. And I'll talk to you about that a little bit more later on. But we're looking at this particular reflex. When you shine light in someone's eye, and we've already seen this happen, and we know parts of this pathway already, when you shine light in someone's eye, the normal response is to have sort of this constriction of the pupil that you see here by activation of specific groups of muscles. And the dilators will work, for example, if you have like too much noradrenaline around, or if, for example, it's really dark out, then this sort of response will be turned on. So this is a really busy slide, and in fact, we've already talked about the pupillary light reflex previously. Do you guys like these types of questions related to the pupillary light reflex? Not really, right? So I'm not going to bother asking you on the term test or on the final exam about the pupillary light reflex. Hopefully, we've already had a question related to this, and hopefully it's something that you'll sort of remember, and it's really to jog your memory. We talked about how this reflex is organized at the level of the midbrain, in a particular area of the midbrain known as the superior colliculus. And this pupillary light reflex actually makes use of the different on-off afferents that exist within your eye that will signal all the way back to the pretectal nuclei. But this actually makes use of the autonomic nervous system. And we didn't talk about it in terms of the autonomic nervous system previously. We just looked at the overall circuit. Now I wanted to just expand on it for your own information and to hopefully get you to think a little bit about the physiology behind why someone's eye might have like a fully dilated pupil. For you to think about physiology not sort of just as an academic exercise, but in real life as well. So if it gets too bright out, we've already talked about this sort of reflex to the third cranial nerve, but it involves the parasympathetic nervous system. And one of the things that it does is it will use acetylcholine being released at the level of the circular iris muscles to cause it to start constricting. If it's too dark, it's actually a sympathetic nervous system response. And because the sympathetic nervous system actually has a very different sort of pathway than the parasympathetic, the sympathetic reflex actually goes from the thoracic cord. So again, think a little bit about this. You are shining light in someone's eye, and or not having light in someone's eye. This reflex actually begins in the thoracic region. It's not anywhere near the head. It's sort of in the lower part of your thoracic region, and it sends another signal all the way back up to the sympathetic chain to the radial muscles that will cause noradrenaline or norepinephrine release in order for the circular muscles and radial muscles to start to dilate. And that's why you get the larger pupil. So if you have a lot of noradrenaline around, these are the muscle groups that are opening up that allow you to be able to see better. And this is why, for example, if you have atropine added or you have other things that interfere with the noradrenergic pathway, you can change the size of your pupil. And things like cocaine, which prevent the uptake of noradrenaline, will cause that because noradrenaline, there's too much of it around, and it's like having a sympathetic reflex response when things are too dark. So just a couple of other things, and we're going to come back to some of these structures later on this evening. When we look at the autonomic nervous system, I told you it's not any different than the somatic nervous system. So in the somatic nervous system, we started off with these reflex arcs. Then we looked at central pattern generators. Then we looked at higher levels in the brain that controlled all these different types of reflexes and different types of locomotion. It's the same idea when we talk about the autonomic nervous system. There are higher brain areas, just like the premotor area, signaling to the mesencephalic locomotory region, which then signals down the different tracts to cause your muscles to contract. It's the same idea with the autonomic nervous system. We can activate different parts of the sympathetic nervous system through higher brain structures like the amygdala, and we'll come back and talk about the amygdala later on. The amygdala is part of the limbic system in your brain. Your limbic system is there to help you develop emotional responses to different situations. And if the amygdala gets activated, this is believed to activate the sympathetic nervous system, especially related to fear behavior. So if something has stressed you out in the past, your amygdala immediately signals that it's a similar situation, it's a stressful environment, activate the sympathetic nervous system, and this is sort of a learned fear behavior. However, you can also activate it not when you're directly seeing a stressful situation or something similar. You can just imagine it. So your cerebral cortex, by just imagining being in the same sort of situation. So remember that term test that you guys wrote and you guys were kind of panicking, and if you relive that and it was really stressful for you, you can actually elicit the same sort of response through the amygdala, which will then activate the sympathetic nervous system. We're going to see the one area in the autonomic nervous system that is really important and is the master integrator and the control system of the autonomic nervous system is the hypothalamus. And when we talk about the hypothalamus, we're going to be talking about feeding behavior, We're going to be talking about circadian rhythms, and these are both two things that I'm going to be asking you about both on the term test and on the midterm. So you should be very clear, because they're really important physiological concepts about what groups of neurons in the hypothalamus, a really small structure by the way, control so many different things like feeding, as well as the circadian rhythms surrounding sleeping and waking up and other types of behavior. I'm going to mention in passing this reticular formation that you have here in the brainstem that is going to also be a pathway that the hypothalamus and others use as well. So your brainstem has a number of different autonomic centers. They cause different things to be cyclical. There is a cardiovascular center that controls the number of times your heart beats per minute. There's a respiratory pattern generator in the part of the brainstem known as the lateral medulla, as well as the pons. And so your rate of breathing that you have right now, about 12 breaths per minute, is set by these groups of autonomic centers that exist within your brainstem. We're also going to not talk too much about this area, which is the periaqueductal gray, but it's like the premotor zone that we have within the motor cortex, the premotor areas, and this is very similar to initiating or calling together groups of autonomic behaviors related to feeding, related to fornication, related to fight or flight. All of this is here at the level of the periaqueductal gray. We're not going to be talking a lot about the periaqueductal gray today. We're going to be looking at the cardiovascular center, within the medulla as an autonomic center. And then in the next few slides, I'm going to take you through some of the ways in which all of these programs can be modulated by a number of different tracks that come out of the brainstem. Okay, so this is not the hypothalamus. We're getting to it. This is sort of the lower end of things, and we're going to be taking a look at this in a few moments. Just out of curiosity, how many of you drink energy drinks? During midterms, final exams, none of you. Wow, what an amazing group. Just coffee, okay. So should I be telling my 16-year-old kid who's been drinking energy drinks to stay up during high school for the last 2 years that she's an aberration? I don't mind telling her that, by the way. Yes, no? Behaviorally, this isn't something that normal university students do. I find that shocking. Really? Wow. And yet they sell them at the cafeteria and everywhere else. So someone must be drinking them. Okay. So why not those energy drinks? Those energy drinks actually have a profound effect on your neurochemistry. And some of those energy drinks have a profound effect on the neurochemistry at the level of the brainstem. And the brainstem autonomic centers that I'm going to get through, some of these are really pronounced in terms of the effects of those energy drinks, which are very heavily caffeinated, and that caffeine actually works on a number of these systems as well. So within the brainstem, we have this loose network of different groups of neurons, and this is known as the reticular activating system. And it's a diffuse, which means that they're kind of clustered in certain regions, but it doesn't run through the entire brainstem. These tend to be modulatory, so they are there to help shift overall nervous system activity. So you could be going from being really vigilant to being really, really sleepy, or not paying attention to being able to pay attention a lot more by activating some of these modulatory systems that I'm going to be talking to you about. So you're going to need to know all of these, because we're going to come back at the very end and talk about these systems again, especially as you're going off to sleep. Hopefully it'll make some sense as to why cholinergic systems will be turning off and why other systems might be turning on. So serotonergic, adrenergic, all of these things, think about what they're designed to be able to do, and think about what you would want. Would these be high when you're going to sleep, or would they be lower when you're going to sleep? So do you need to be paying a lot of attention while you're sleeping? Again, this will be really important in this sleep-wake cycle. And I'll talk about the pontine system in a moment. Serotonergic, again, it's used in stressful situations. If you're sleeping, I'm hoping it's not a stressful situation for you. It also influences mood. It's also involved in the sleep-wake cycle. Adrenergic, when you need to really be vigilant for that fight or flight response, this adrenergic system is really important for stress. And these last two, serotonergic and adrenergic, have a number of very, very strong motor effects. And we're going to take a look at some of these motor effects as we go through the next few slides. I don't need you to memorize everything on these slides. All of these slides and all of these systems are related to these groups of neurons, this diffuse system of neurons that are found in the brainstem. So this is the brainstem, and you can see that whether we're talking about the serotonergic system, which has a number of different nuclei, so these larger blobs that you see here with arrows coming out of the blobs represent the diffuse groups of neuronal cell bodies in the brainstem, or we have one that is responsible for acetylcholine that's coming out of the brainstem. All of these are ones that I want you to be paying attention to more for the anatomical location and the nuclei that are associated with them than the actual topology of where they spread. You can see that all of them, every last one that I'm going to show you, they might have a small concentration in the brainstem, but their effects are throughout your entire brain. That's why it's CNS modulatory. It modulates everything within the CNS. So when we talk about acetylcholine, we're talking about this reticular formation that is found in the pontine region, and that was on the previous slide. And this is the group of neurons that will form and release acetylcholine throughout all of these different parts of the brain. It will target the thalamus, it will target the basal nucleus of Maynard. All of these different regions will be influenced by acetylcholine because it starts here at this pontine reticular formation. And that is the acetylcholine group that I want you to make sure that you are aware of, this group right here, the pontine reticular formation. When we talk about serotonin, and again, you don't have to worry about its effects on mood. All of this is sort of just extra information for you if you are really interested in this system, is the anatomy of the raphe nuclei. And each of these are the raphe nuclei. This is the structure known as the brainstem. And you can see that it spreads out to a number of different areas. In this particular case, also into the cerebellum. So you can see that the acetylcholine system, for example, doesn't go into the cerebellum, but the serotonergic system does. And again, that's the major things that I want you to take away. The norepinephrine system starts off at the locus ceruleus, or the blue location, or the blue group. If you took a brain and you were to slice a sagittal section, as you see here, and you were to take a look at it with the naked eye, you would be able to see this group of neurons being without any staining, showing up as a light blue or pale blue color. And this locus ceruleus is where we find the origin for all of the norepinephrine system. So this forms and releases norepinephrine throughout the entire brain that you see here, including to the level of the cerebellum. And again, I won't be asking you if you're wondering about specific targets that you see here and specific targets that you see here. It's really the origins of where these different systems come from that I want you to be more concerned about. So the histaminergic is the last sort of modulatory system that we're going to take a look at. So this is originating in the tuberomammillary nucleus as part of the hypothalamus. We'll see this again later on this evening when we talk about how you go to sleep and how you wake up. This histaminergic system, again, spreads out throughout the entire brain, and it is really important when you need to stay awake. Sorry, I'm just trying to get the laser pointer back. When you need to be able to stay awake, you need to have a lot of this histamine around. This histamine has to be interacting with a number of other systems. It will also help to modulate acetylcholine. It will help to modulate a number of other neurotransmitters. How many of you have allergies at this time of year? What do you take for your allergies? So you take antihistamines. What's one of the side effects of antihistamines? So drowsiness, right? Because the antihistamines that work on your allergies and for prevention of degranulation of some of your mast cells that cause all of the things that cause you to sneeze, etc. It also works on your brain. So it blocks all of the histaminergic receptors in your brain, in your gut, as well as on your different immune cells. And so if this is important in you staying awake, when you block histamine's actions, you are going to be falling asleep. And so we'll see how this histaminergic system is important in a few slides. So as I mentioned to you earlier, we're not going to be talking a lot about the periaqueductal gray. And the periaqueductal gray is one of the regions that is involved, similar to a premotor center, almost like a central pattern generator is, for organizing different types of autonomic nervous system behavior, whether it's related to feeding, fear reactions, etc. All of this is regulated at the level of the midbrain, at the level of this periaqueductal gray, and this is in some ways, as on that previous slide, is related to it being like a premotor center. So it doesn't actually set the rhythm, it doesn't actually cause it to be selected, but it helps to organize these different types of autonomic behaviors, things like feeding, things like fear responses, things like fornication, etc., that are really important at a lower level. So I'm going to talk to you about specific autonomic reflexes. These will be making use of both the sympathetic as well as the parasympathetic nervous system. The enteric nervous system that we're not going to be concentrating on can also function independently of sympathetic innervation or parasympathetic. You'll learn about that later on when you talk about the digestive system. Simply having food within your gut actually causes better contractions. You don't need the parasympathetic causing it to contract more. And that's because within the walls of the digestive tract, we have a number of different receptors that will tell if the gut is being expanded by having food within it. So I'm going to go through a couple of different autonomic reflexes. The ones that I want you to pay attention to, I'll point out to you. These are reflexes that are not any different than the motor reflexes that we saw related to your muscle spindle stretch, Golgi tendon reflex, any different than any of the other reflexes that we looked at earlier. There has to be a sensor, something that detects something going on. There has to be the transmission of that information, and then there has to be a response, and that's what a reflex arc is. So for example, in this particular diagram, if for example you saw that there was an increase in blood pressure that's detected by these pressure receptors called baroreceptors in the carotid sinus, it would detect that there's too much blood pressure. Too much blood pressure for prolonged periods of time is not really good, so it will signal, the baroreceptors will signal to the brainstem as part of an autonomic reflex. It will activate the vagus nerve, which is your parasympathetic nervous system, and the parasympathetic nervous system will then act on the ganglia in the SA node to overall decrease the heart rate. So just to make sure that you are paying attention, and again, the details of this not being so important. This is a really simple reflex just to show you that it's not very different than the reflexes we saw earlier. If this is the parasympathetic nervous system, what is the neurotransmitter that is being released by the vagus nerve, which is a parasympathetic fiber at the SA node? So it is acetylcholine, perfect. And again, this will act to lower the heart rate. So blood pressure was too high. Reflex activity is to cause it to slow down and to reduce the blood pressure as a result. The sympathetic reflex that you see here, if for example the blood pressure was too low, also detected within the carotid sinus, one of the things that would happen is it would signal back to the brainstem. This is different, it's sympathetic, so it's not at the level of the craniosacral, which is parasympathetic. It will send a signal down to the thoracolumbar region, and from the thoracolumbar region, it will then signal through those nearby sympathetic ganglia to eventually cause the sympathetic nerve to innervate the heart to release its neurotransmitter. And what is its neurotransmitter that's being released? So this again is norepinephrine, noradrenaline, or the epinephrine system to increase the heart rate because your blood pressure was too low. So there's always sort of a reflex that's built in This is a really simple one that allows you to compensate for when something is going out of whack. When you are facing something that physiologically is changing, how do you get things back into homeostasis? We use these autonomic reflexes. So these are ones that I'm going to go through related to the hypothalamus and different parts of the midbrain. And there are some that I'm going to ask you to know more than others. So for example, the one that is involved in blood pressure, in particular the one that involves this nucleus tractus solitarius, which is this NTS. This is one that I'm going to ask you to know. I'm going to cover it in some more detail in the next few slides. We're not going to go through the retina again. I already told you that the pupillary light reflex related to cranial nerve 3, related to adrenaline release or acetylcholine. I'm not going to be testing you on that again because I already have tested you on this. So the retinal responses, I don't need you to know. I'm just going to point out for the time being that the suprachiasmatic nucleus is part of this hypothalamus, and we're going to see that this area of the hypothalamus is going to be really, really important when we are talking about how you go to sleep, how you wake up, and circadian rhythms. So again, this is the hypothalamus as a reflex related to a really complex type of behavior. We're not going to spend a lot of time on the olfactory and limbic systems, and we're going to talk in the next slide about the reticular formation, which eventually will relay information up into the hypothalamus. So there's a lot of things on this slide, and some of this may be a little confusing to you. So let me point things out as we go along. So your body is always trying to determine like how warm you are, how cold you are. So you have all of these different thermoreceptors on your skin, and we've talked about these thermoreceptors on your skin. You also have thermoreceptors inside your brain. And these ones that are there at the level of your brain are somewhat similar. They're always sensing the body temperature, especially as blood is flowing by it. And it's trying to determine how is my core body temperature doing related to the temperature outside on those thermoreceptors on the skin. It's comparing that information all the time. It's like checking Facebook all the time to see what the status updates are like. Yeah, things are going pretty good. You know, the Golden State Warriors just won Game 7. Way to go, Stephen Curry. Really like that. And it's always kind of looking at the difference between what's going on internally versus externally. There are thermoreceptors that are at the base level of the hypothalamus. These are also detecting changes within the environment. Now, I told you earlier, related to your skin, that we have more of the thermoreceptors related to cold than we do to heat. Within your hypothalamus, however, within your brain, it's the exact opposite. So we actually have the warm sensitive ones that are outnumbering the cold sensitive ones. And these are the ones that are ultimately going to control whether or not you engage in thermogenesis. Do you need to increase body heat? Do you need to decrease your overall body temperature? So for example, if you are too cold, one of the things that these different hypothalamic neurons do, they will signal for skin vasoconstriction, so you have less blood flow to your skin, which means less heat loss through your skin. It will cause the hairs on your body to increase, to capture as much air as possible, to cause more body warming, so this piloerection. It will also decrease cardiac output, because the more blood that you have flowing through, going out to your periphery, the colder you're going to be feeling, so decreasing your cardiac output and the amount of blood flowing throughout your periphery will also decrease because you're feeling too cold. You're going to be moving fluid out of your plasma back into your cells. You're going to be engaging in a shivering response, which is mediated through the posterior part of the hypothalamus. And all of this is going to be increasing adrenergic input as well. So if you're too hot, the exact opposite happens. Different part of the hypothalamus is activated, the anterior hypothalamus, and this relies a lot on serotonergic input. So these areas of the hypothalamus are different. So we have posterior parts of the hypothalamus, anterior parts of the hypothalamus that respond when you're too hot or when you're too cold. They rely on different types of modulation, so whether it's serotonergic input that is important or adrenergic input when you're feeling cold, that's important. These are differences in the hypothalamus depending on the temperature that your thermoreceptors in your hypothalamus are able to detect. So I'm just going to go through this one sort of regulation that wasn't mentioned earlier. Oh, actually, I did mention the nucleus tractus solitarius earlier. And again, this becomes more anatomical than anything else. So the anterior and posterior parts of the hypothalamus do exactly opposite types of things when you're too cold or when you're too hot. And this part of the brainstem will do something very, very similar. Just out of curiosity, when I mention things like rostral or caudal, what do those words mean to you? Rostral, caudal? It's all this. It's all this. Okay. So rostral does mean toward your nose, and caudal means toward your tail region. So in other words, if you're looking at it from the nose end to the tail end, it just gives you some directionality within the brain. So if we were to take a look at the medulla, right beside the respiratory center that controls your breathing, there is a center that controls your heart rate. It controls your blood pressure. This is the blood pressor. So every time, for example, you are standing up or lying down, your blood pressure changes. You don't faint or you don't have a stroke because this cardiovascular center has an autonomic reflex associated with it. You don't have to think about it. Your autonomic nervous system doesn't get activated through a long, complex thing. It's a reflex arc that takes place. And this controls like your overall blood pressure. And this is the cardiovascular center that's found in the medulla, very close to the respiratory center. And the group of neurons that are important in this cardiovascular center is this nucleus tractus solitarius. And this is the nucleus tractus solitarius that you see here. And it receives a number of inputs from baroreceptors. Remember the carotid sinus that I showed you earlier, blood pressure being detected from the carotid sinus. Those baroreceptors will send information to the nucleus tractus solitarius, and it has to do something with it. So this is an expansion of what we looked at earlier. So one of the things that it does is it will send information first to the caudal part of the ventrolateral medulla, or the VLM. So this is the VLM. It will send information to one part, the one near the tail region, the one closer to the bottom of the medulla, closer to the spinal cord, the caudal end. It sends information here first. And one of the reasons why it has this particular orientation is that the two different regions, the caudal region of the ventrolateral medulla, so it's a long group of neurons, so it's a long nucleus, the one near the tail end, the caudal end, and the one near the head end, they have very, very different projections and different functions. So within this ventrolateral medulla, a long group of neurons, the rostral half, the one that's near the head end or closer to the nose end, actually excites sympathetic efferents. And its main function is to increase the blood pressure. And we say that it has a pressor function. The rostral end, in other words, elevates your blood pressure if your blood pressure is too low. But the information goes first to the caudal half. And that's because the caudal half actually has the function of inhibiting the rostral one. So it actually controls whether or not the rostral region gets turned on. So it can decide if the blood pressure is going to be too high, that it will inhibit the rostral one. So the rostral is there, it's increasing blood pressure. Now we've detected that the blood pressure is too high, it will turn off this rostral half. And we say that the caudal half actually has a depressor function. It lowers your blood pressure rates. So this together goes back and tries to explain in some more detail what we looked at here with this very simple autonomic reflex. And I'd like you to make sure that you know about the caudal half versus the rostral half in the ventrolateral medulla, that this is the area of the medulla that is responsible for elevating or decreasing blood pressure. I'd like you to know where these signals are coming from, which is the nucleus tractus solitarius, and how these two things work together, like how the caudal and the rostral halves are working together. And these other structures that you see here are not ones that you should be worrying about, although we've talked about the raphe nuclei previously. Any of you happen to remember raphe, where we've seen it? A few slides back, the modulatory systems. Remember which neurotransmitters, raphe nuclei? So we're gonna do exercises related to increasing your longer term memory. I think it's really important, but it's something that we'll be coming back to anyway. So we'll talk about that in a few moments. Okay, so going back to the hypothalamus, and this slide you definitely need to make sure that you go back and review. You should make sure that you know about temperature regulation. And again, I'm going to go through the control of eating and homeostasis in a few moments. Your hypothalamus is that master regulator. It is there to make sure that all these autonomic programs are keeping you in a state of homeostasis, which means that your blood glucose levels have to be at the right level. Your blood pressure has to be at the right level. All of these different functions. It's all mediated here at the level of the hypothalamus. And if something is off, it will activate the autonomic nervous system, it might activate the endocrine system, or it might actually involve activating the somatomotor systems for eating, etc. And all of this is sort of related to what I want you to be concentrating on the next few slides. So I told you earlier on that when we get to these higher order structures, that within the hypothalamus, we're going to be talking about things related to feeding behavior and also related to your sleep-wake cycle. This is one of those groups of neurons within the hypothalamus that I want you to make sure that you are very, very comfortable with. I can guarantee you that you will be asked this particular group of slides, this one and the diagram that comes along with it, both on your term test as well as on your final exam. It is a really important series of slides because it is a foundation within physiology that you need to know what regulates feeding. It is one of those circuits, it's one of those things that we understand incredibly well because it also intersects with endocrinology, digestive system, the brain, and everything else. So if we're to look within the hypothalamus, we're going to see a couple of different nuclei. And I'm going to outline these nuclei first on this particular slide, and then I'm going to come back and show it to you in a diagrammatic way in the next slide. So we have one group of neurons which are known as the arcuate neurons, or the arcuate nucleus. Within the arcuate nucleus, we have two opposing sets of neurons. We have ones that want you to feed more, and then the other group that want you to stop feeding. And these arcuate group of nuclei have neuropeptide Y, or NPY, and these groups of neurons through the paraventricular nucleus in the arcuate nucleus controls feeding behavior. There is another group within the arcuate nucleus that is melanocortin containing. The neurotransmitter there is melanocortin, and when this is activated, it will activate a hypothalamic program that will suppress feeding. So in other words, within the nucleus, you have both feeding and the cessation of feeding behaviors through two different types of neurons. One is neuropeptide Y. You stimulate neuropeptide Y, you're going to eat. You want to stop eating, then you're going to activate the melanocortin group of hypothalamic neurons. There are a couple of modulators as well. So this is one group, and then this other group, the ventromedial nucleus, contains receptors for blood levels of glucose. And what it will do is if the blood levels of glucose get higher and higher, it will start inhibiting the neuropeptide Y containing receptors. So a lot of other factors, including things like leptin, which is secreted by the white adipose fat tissue. If you have more and more leptin, it's because your fat cells are getting bigger and bigger, which means that you are feeding better and better. leptin will act to stop some of these neurons as well, and will inhibit these NPY-containing neurons. So diagrammatically, it looks like this. So what we're looking at is this structure here. This is the hypothalamus. This whole structure here in white is the whole hypothalamus. The hypothalamus is always drawn this way in every sort of textbook. And right down the middle of the hypothalamus is this third ventricle. So we have cerebral spinal fluid flowing through here. And then we have all these neurons sort of sitting on the edge of this third ventricle. This is the arcuate nucleus. This whole structure here is the arcuate nucleus. Within the arcuate nucleus, we have two opposing groups of nuclei. We have this group of nuclei that you see here, and this group of nuclei are the ones that contain the neurotransmitter NPY. And NPY, when you stimulate this group of neurons, it will cause you to start feeding. It wants you to help, it wants you to start eating, and it will signal through the paraventricular nucleus to start eating. There's another group of neurons also found in the arcuate nucleus that contain the melanocortin, and this is the abbreviation for melanocortin that you see here. And when it gets activated, it does two things. So one is it inhibits the NPY-containing group of neurons. So when you're feeling full, in other words, you want to stop eating, it sends a signal over to inhibit the NPY-containing neurons, and it will also signal to another area of the brain to say that you are full. And this feeling of satiation, where you feel like you're full, gets turned on by the melanocortin. So this other group of neurons that you see here, the ventral medial nuclear neurons, are the ones that can also put a brake on this particular pathway. So again, this is the one that will cause you to eat, the NPY or neuropeptide Y containing neurons. And this group over here, as you start eating, and this process as you start eating, as your blood glucose levels go up higher and higher, and it's really your the drive for you to eat actually is where you have a drop in your blood glucose levels. You know those commercials that have Snickers and feeling hangry? It's actually true. So for you to feel hunger, your blood glucose levels start dropping more and more. It signals to the endocrine system, your stomach starts growling because of your low blood glucose. You start releasing things like ghrelin, and I'll get to this again in a few moments. But this blood glucose is the major influence on hunger. It will, as you start eating, your blood glucose levels come up. The blood glucose levels, as they come up, are detected by these neurons here within the ventral medial nucleus to then signal to the NPY-containing neurons to stop eating. You are full. You don't need to have as much glucose around, and this is where you should stop, and this is where this group of neurons is no longer being activated. So there are some other factors that will also play a role, not just glucose. We have PYY, we have ghrelin, we have leptin. All of these can act on different groups of arcuate-containing neurons. So ghrelin, when you're hungry and your stomach is growling, every time your stomach growls, your stomach is actually releasing a substance known as ghrelin. This ghrelin then signals up to your brain through nerve fibers to activate this group of NPY-containing neurons. PYY also activates and turns on this group of neurons as well. And leptin, when you have too much of this, because you have a lot of leptin being released from the white adipose tissue, will actually turn on this group of neurons here that contain melanocortin. So we have a number of sort of feedback between the endocrine as well as the other systems. So this ghrelin is a hormone from an empty stomach that is orexigenic. So I'd like you to hold on to that word for just a few moments, and then we'll take a break. Orexigenic means feeding behavior, and orexigene is a feeding hormone. So this is released from your stomach. It acts in a positive way to cause you to want to feed. PYY is released from the intestine in response to food. It also inhibits this feeding behavior. CCK, OEA are other fatty acid derivatives, and it will also inhibit this feeding behavior as well. So I'd like you to make sure that you understand this regulation of feeding, the circuit behind the feeding behavior that you see here, and then the feedback control from the gut that controls feeding behavior as well, and how they regulate this feeding. And then we'll come back and we'll talk about some of the other pathways after the break. Just out of curiosity, borborygmus, any of you have heard of this term? What is a borborygmus? So it causes the release of ghrelin. So when you hear that growling sound, that sound is called a borborygmus, and that borborygmus is the thing that actually causes ghrelin to be released from cells within your stomach, and that's sort of one of the reasons why I sort of have this up here. So let's take a break now, then we'll come back and we'll finish off today's lecture. Yeah, it's not a true sympathetic response. That's sympathetic local sympathetic. You're correct on the line for we need that so ghrelin for sure is positive feedback. So this is
Okay, yeah, because this is saying any other disease it's like a food hit, so then it needs to stop.
Simple to ingest some kind of another important derivative as like a horrible dietary thing. Yeah, people are yeah, loving the target.
oh, someone actually they may show up
Really?
right?
Just about this slide, I think you mentioned that if these neurons are activated, because I think our Q8 neurons, right, are just here, and
Okay. Let me Okay, if you don't mind, I'm going to get going. Couple of questions actually came up during um, the break that I actually thought were relevant, and I wanted to go back and kind of talk very briefly about them. These are different factors. This is sort of a summary diagram that actually has a number of factors that are put on the same slide. They can all work together in this way, for example, but for you to understand it better, it's probably a wiser idea to just go through one pathway at a time. So glucose is only working on the ventromedial nucleus, and it's only working on neurons here. This is working to inhibit this particular group of neurons that contain NPY, and it's better to kind of make sure that you're aware of that. The other thing as you're studying, because it becomes really important, is that there are a number of different nuclei that are put out here for you. So this arcuate nucleus actually contains two different types of neurons, ones that help you eat and ones that tell you when you're full. They also signal to different groups of neurons up here. The paraventricular nucleus is another group of neurons that you see here, and the PVN is different than the groups of neurons that you see here. Please make sure that you are comfortable with where you find different types of neurons in the hypothalamus. This whole structure up here in white is the entire hypothalamus, and then in gray up here in this white region, what you're seeing are specific groups of neurons, and we call these groups of neurons nuclei, and make sure that you're clear on that. This diagram down here on the bottom becomes really, really confusing. So ghrelin stimulates the NPY neurons to make you increase feeding. PYY, does it do the same thing? You should be following what is on the bullet points here. So you should be linking the bullet points here with what is on that previous diagram. And we also have things like CCK or cholecystokinin and OEA. And I won't try to pronounce what OEA is. This is down here for you. That will also work in ways to inhibit feeding behavior. And this is the diagram that you should be more comfortable with. Any questions or things that I can clear up for you now before we go through the last sort of section? Yeah. Um, so the question, if I understood this correctly, the glucose, so you start eating because you're hungry, your blood glucose has been low, you start eating, and as you start eating, the food that you have will start increasing your blood glucose levels. And as your blood glucose levels rise, this is all part of the bloodstream that is flowing by. As your blood glucose levels rise, the glucose is detected by these glucose receptors in these neurons. And they will in turn signal to the NPY containing neurons to stop eating, right? So as your blood glucose rises, it's another signal to stop eating. As you start eating, you're also going to be releasing more PYY. It's also going to stop you from eating. Um, Leptin will also activate this pathway to stop you from eating. Any other questions? Okay, so I'm going to go through the amygdala as part of the limbic system really quickly. I'm not going to go into it in a lot of detail. I know many of you have heard about the amygdala in different courses, like in psychology. This is actually part of the basal ganglia. It is really important for autonomic behavior. We've seen that earlier on in today's course, and it receives input from parts of the limbic cortex and from the limbic cortex that is very close by or in the border region of the amygdala. And one of the things that it does is it will actually have output onto some of those different autonomic brainstem nuclei or functions that we saw earlier. Generally, when you activate the amygdala, you turn off parts of the periaqueductal gray and hypothalamic autonomic systems that we saw earlier. So if it's normally there, if the amygdala is normally there to act as a brake, one of the things that's been found is that lesions of the amygdala can actually result in uncontrolled autonomic behaviors. Amygdala is there to act as a brake for autonomic behaviors, and now the amygdala is gone. What happens to all those autonomic behaviors? Literally, they become uncontrolled. So you have a lot of autonomic behaviors like sham rage, so including like being really aggressive, sham rage, hypersexuality, etc. Have any of you heard of these different types of disorders? Do you know how they occur by any chance? How do you damage the amygdala? So the amygdala it's about the size of your thumb's fingernail. It's found on the border of the limbic cortex. It's very near your hippocampus. How do you actually damage something in the medial temporal lobe? You could damage it through stroke, possibly. Anything else? So stroke is definitely one way in which the amygdala can get damaged, but again, you have two of them. And you have to lose both of them for this to occur. Anything else that you can think of? So certain drugs can inhibit the amygdala as well. Um, and do you have any idea from any of your psychology classes why I have a picture of the Eiffel Tower? Sorry? The two legs are the... Okay, I think you're reading too much into it. Okay. Um, so imagine, for example, the limbic cortex, the things that actually control fear responses, and it's one of those things I mentioned about fear. How would your life look like if you weren't afraid of anything? I know it's really hard to imagine because you're here at U of T, you're afraid of everything, right? So I mean, is that going to be on the test or is that not going to be on the test? But imagine you had a life where you had no fear of anything. What would your life look like? Your life would look a lot like this. In fact, this is one of the individuals who is documented as having bilateral removal of the amygdala. Again, we won't go into all of the details around Erica Latour-Eiffel. She actually had no fear, and so she didn't really care what other people thought, and so she ended up marrying the Eiffel Tower. Um, and again, she goes even further. She did not only marry the Eiffel Tower, she had sexual relations with the Eiffel Tower. She also, I kid you not, you can look it up when you go home. Um, she decided that the Eiffel Tower was not enough for her, so she decided to have hypersexual relations with a bridge that was nearby as well. So this is what your life would look like if you did not have um, the amygdala, sort of part of the limbic system that is controlling your autonomic behaviors, those pre-programmed behaviors at the level of the periaqueductal gray and the hypothalamus. If you have no fear, you don't really care what people think about you, right? And again, you make use of those different programs that are in there. So the amygdala is also really important for emotionally laden types of memories. The best memories that you have, or the ones that last the longest, are the ones where you have different emotions associated with them. Oftentimes we tend to think of them as being like really, really scary. So if you've been scared once about something, you're likely not going to do something again because it makes such a long-lasting memory, again, involving things like the amygdala. Um, Again, the amygdala, for those of you who have taken other psychology courses, we don't store memory in the amygdala as far as we know. Again, there's some debate about whether or not the amygdala is capable of memory storage. We think that memory associated with emotion is distributed throughout the entire cortex and associated areas of the cortex and the limbic cortex. But it's really important for learning about different types of behaviors, autonomic behaviors, or other types of things that are involve emotion related circumstances, as well as, we think, helping you to learn things in the appropriate sorts of ways. And this gets into this whole idea of the limbic cortex. And this is sort of the edge of the limbic cortex. So it's kind of broken up into pieces. This is the cerebral cortex out here on the periphery in gray. And in the darker gray, you actually have the old parts of the brain. So for those of you who have taken neuroanatomy, the old parts of the brain are located deeper in the brain. And the old parts of the brain include things like the cingulate gyrus, the insula that you see here, and part of the hippocampal formation, and parts of this periaqueductal gray that you just see in the midline. And all of these different things are believed to be really important, phylogenetically very, very old. This is probably the first parts of the brain as you take a look at it evolving, and these are the phylogenetically oldest parts of the brain that we have. And all of these different areas in the brain, things like the insula signaling for disgust, or the cingulate gyrus being involved in different forms of memory, we think that these are all important for motivation related to memory, memory consolidation pathways, etc. And we know that neurons that are found in these different parts of the limbic cortex, and including the hippocampus and parahippocampal gyri, will also project and send projections to the hypothalamus, the periaqueductal gray, and also to the amygdala. So there's sort of a trade-off. There's also some signaling going back to those limbic areas as well, and to the amygdala as well. And we think that this is really important for your memory. And I don't know if Dr. Key talked to you as a group about mechanisms that underlie learning and memory, but these are things that are, again, really important in terms of studying, thinking about how to make your learning, as you're trying to make some sort of emotional context to your learning. If you can find a way to do it, whether it's through music and activating these different emotional responses, this limbic system, your memories will last a lot longer. And it's up to you to figure out what sorts of things really get your emotions going in the appropriate ways to help you to learn, because there's always this fine balance. So I'm going to spend the last little bit talking to you about two different things, circadian rhythms and the structures associated with circadian rhythms, and then some other things related to those circadian rhythms, including ultradian rhythms, etc. So along the way, we're going to be looking at what areas of the hypothalamus are involved in different hormones and neuromodulators being secreted and formed, how different types of behaviors like sleeping and waking are being modulated, and how all of these different circadian rhythms get modulated to help you stay alert at the appropriate, how it changes your mood, and how it even affects your affects things temperature. So most of you will have heard of circadian rhythms, and because it's very much like the autonomic patterns that we saw before. Remember, we have central pattern generators in your spinal cord, in your brainstem that work on this oscillation. We have the same thing in the hypothalamus. The hypothalamus, again, is an autonomic center, but it also has oscillations. These oscillations occur in 12-hour cycles that add up to 24 hours that allow us to do certain things during the day, certain things at night. And this is entrained in us because we have visual input, ganglion cells that contain that melanopsin that I spoke to you about earlier. Those melanopsin-containing retinal ganglion cells projections to the lateral geniculate nucleus, which is what we learned about earlier, and then into the visual cortex. Retinal ganglion cells will send their axons and their projections to an area of the hypothalamus known as the supranucleus, and then they will also send projections to a pretectal area through sympathetic efferent. They will cause activation of what's known as the pineal gland. So these retinal ganglion cells, the melanopsin ones, have two different pathways that are really important to us in understanding sleep and circadian rhythms. One pathway is through the suprachiasmatic nucleus that you're going to see abbreviated as SCN, and the other one is through the body and the formation of things like melanin. So here are some of the different structures. This is the suprachiasmatic nucleus. This is sort of where the optic chiasm exists, and this is the group of neurons that we're going to be taking a look at. So this suprachiasmatic nucleus is right above that optic chiasm, makes it in perfect location. Remember, you're seeing things through the retinal ganglion cells, they're detecting light levels, they're helping your brain know when it's light and when it's dark. It's perfect, perfectly situated right above the optic chiasm at the level of the hypothalamus. Again, very similar. What other groups of neurons do we find here in the third ventricle area related to feeding? So if I were to talk to you about feeding behavior, remember the arcuate nucleus. So this is, again, third ventricle. It's a little bit more anterior, but it's also in the same general vicinity. And so this is the hypothalamus as well. The activity in the suprachiasmatic nucleus neurons is lowest at night and highest during. And it undergoes a number of different oscillations where different genes are being turned on at different times and different genes off at certain times. And this is why we say it has a 24-hour oscillation. We won't be going into all of those different types of genes and their transcription and translation products like the BMAL1 as well as the periodicity genes being expressed. It's just to give you an idea why we talk about this 24-hour clock. It's related to gene transcription, has little to do with direct light levels overall. It starts the process, but this is how we determine it's a 24-hour clock. This gets modulated by retinal input, pineal input, which varies from season to season, and also the amount of serotonin that comes in to this suprachiasmatic nucleus. Another area that I mentioned that was really important was this pineal body, and this pineal body is also at the very top of this third ventricle, and what its major function is to secrete melatonin into the cerebrospinal fluid as well as into the bloodstream. Okay, so we haven't talked about the function of the suprachiasmatic nucleus just yet. We've talked about where it's located, and I've just told you the function of this pineal body. And this pineal body, its main function is to produce and secrete melatonin into the bloodstream and into the brain or into the CSF. Any questions about this before I go on? Please make sure that you're aligning and correctly identifying that melatonin is being secreted by this pineal body. So I told you that the suprachiasmatic nucleus is responsible for a really big behavior. It's autonomic because it's in the hypothalamus. And this sleep-wake cycle is actually involves a number of different structures within the hypothalamus. There are two different centers that you need to be concerned with and understand in the hypothalamus related to the sleep-wake. So again, make sure that this is okay for you. And I'm going to go through this slide. It's also related to the slide that comes right after. So you should be able to apply and identify these concepts on the next slide that's immediately after. There is a group of neurons that are involved in the initiation of sleep. And this occurs in the ventrolateral preoptic nucleus in the hypothalamus. So one of the things that activates this group of neurons in the hypothalamus that starts you off in the process of sleep is the circadian clock, that 24-hour oscillation. If you are in sleep debt, where your brain is very, very low on glycogen stores and very, very high in adenosine levels, then you are in sleep debt, as well as other inhibitory signals. So do you guys, have you heard of sleep debt before? Yes? How many of you have pulled an all-nighter? Okay, so many of you have. Hopefully Monday evening before next class, you'll be pulling an all-nighter as well. What happens when you pull an all-nighter? Nothing? So you really have to pull an all-nighter. What happens to you after you've pulled an all-nighter? Yeah, you kind of feel like you're going to crash, right? And a lot of you will crash after you've pulled an all-nighter. This is because you are in sleep debt. Because you've been staying up and because you've been suppressing this sleep switch within the circadian rhythms that activate the hypothalamus, one of the things that happens is your glycogen levels drop. And because glycogen levels are dropping and you are using up, you're using energy and operating the neurons in your brain, the adenosine levels within the synaptic clefts are also going to be increasing. So one of the things that adenosine does is it helps to actually put you to sleep. Caffeine, by the way, which helps to keep most of you awake, not all of you. Caffeine won't work on all of you. If I were to guess, about 20% of the class, if you took coffee or tea and it had lots of caffeine in it, probably has no effect on you whatsoever. And you're caffeine insensitive. But for the majority of you, if you take caffeine and it keeps you awake, it's because you're blocking the adenosine receptors. So even if there's lots of adenosine around, caffeine blocks the receptors, and so it prevents you from feeling as tired. But eventually it'll catch up. No matter how much coffee you drink, at some point your sleep debt will catch up with you because the adenosine levels just get too high. There's another switch that is also at work within the hypothalamus. This is called the waking switch. This is run by the tuberomammillary nucleus that we talked about earlier. So I mentioned modulatory neurotransmitters. Do you guys remember, well, it's written here for you. So that's just the histamine-containing group of neurons. It starts off here in the TMN, and this is excitatory to the brainstem activating system. So when this system is on, it's excitatory. It turns everything else on within the cortex. If you turn the other system on, it is generally an inhibitory signal elsewhere. And just like with the central pattern generators in your spinal cord, in your hypothalamus, when one is on, it turns the other one off. They are mutually inhibitory. Yeah, I know, right? Exactly. So hopefully that's something that you will remember from your spinal cord lectures related to the step cycle of locomotion, the two half centers. You should make sure that you're spending time going over those. On Friday, I'm going to go through that again with the group that shows up, and hopefully you'll remember about this mutual inhibition as sort of a property of the intermediate zone neurons. So this is the hypothalamus. And this is the TMN that is responsible for waking. And this is the ventrolateral preoptic neurons that are responsible for sleep. So these are two different groups that are mutually inhibiting each other. So this sleep group of neurons is inhibiting the waking ones when it's active, and when the waking ones are active, then they are also signaling to turn off the ones that are involved in sleep. There is another group of neurons that we haven't really talked about, this suprachiasmatic nuclear group of neurons. So it will signal to the VLPO, or the ventrolateral preoptic nucleus neurons, to initiate the sleep pattern, but it will also signal here as well to the TMN. So your suprachiasmatic nucleus signals to both groups, one for sleeping and this other group that is involved in histamine release that we're going to talk about in a few moments. And this group over here, the orexigenic group of neurons, is going to be signaling to the histaminergic that you see here. So I'm going to take you through the next few slides that go back and talk about this. So it's sort of in the reverse order. And I'm going to be talking to you about this orexigenic group of neurons and the TMN and why these are important. So this sleep-wake cycle that we're talking about, the waking center, the TMN group that provides all of the histamine for the rest of the brain and project all over the brain, this is what's involved in waking you up. And we know that there is a reciprocal inhibition between the sleep center in the VLPO and also this group of neurons here. But we really haven't talked about the regulation, like what begins this whole process and how do we actually go from a sleeping state to a waking state. And this involves that group of neurons that I just showed you. This is orexin. It's also known as hypocretin. Two different groups at the same time in the early 2000s were in this amazing race to figure out what this group of, what this new neurotransmitter that they had isolated, what was it responsible for? And they were both trying to publish. They came out and published within 1 week of each other. One group called it orexin, the other group called it hypocretin. But they were both right. They both found exactly the same thing. These neurons that they published in Cell at the same time, this group of neurons in the dorsal hypothalamus gets activated or turned on by the suprachiasmatic nucleus. And its main function is actually to turn on or excite the histaminergic neurons that are found in the TMN. So this orexigenic or orexin-containing group of neurons is really there to help you become awake. So it receives input from the suprachiasmatic nucleus. This group of neurons gets turned on. In turn, it activates the histaminergic system within the TMN, and it helps to stabilize the waking state in relation to the circadian rhythm. So remember, when the TMN is on, and now you've turned it on, it will turn off the VLPO group of neurons. We know that this group of neurons is really important in helping you stay awake. So for example, if you are an individual who has narcolepsy and or cataplexy. What is narcolepsy? Just out of curiosity, I know some of you might have heard of it. Narcolepsy? Yep. Yeah, so narcolepsy and cataplexy are related when you will at very inopportune times just randomly fall asleep. And you randomly fall asleep, even during the day, not like you normally would during the evening, because you don't have these orexin-containing neurons. Your brain has developed antibodies which destroy these neurons, and over time you lose orexin. You lose orexin, you can't activate the TMN, and as a result, the sleep centers are actually activated much more powerfully, and that's one of the issues that occurs in narcolepsy. Again, it also provides a circadian drive for feeding. After you wake up, usually you start eating, and one of the reasons why you start eating and where the name comes from, orexin is actually a feeding enzyme, and orexin is usually highest or most active when you've woken up after you've activated TMN, and this is usually a drive for you to start eating as well, and this is through the paraventricular nucleus. So I'm going to take you through a couple of things. You don't have to memorize what's on the next few slides. I'll go through them really quickly with you. I know that many of you have already had these. If you start looking at the different stages of sleep, many of you will have heard of rapid eye movement sleep. Some individuals suggest that it's during REM that you might have dream episodes. It's not entirely clear, by the way. There's still a great deal of debate in neurobiology and in neurophysiology whether REM sleep is where you actually dream or not. In some ways, this is thought to occur in REM because the brain rhythms that you generate in REM are similar in some aspects to the waking state. So, if you're thinking about what you're doing during the waking state, and if your dreams mirror what's happening in the waking state, REM has been thought to be associated with dreaming. Light sleep are stages 1 and 2 that I'll show you on the next diagram, and then deep sleep, also known as slow wave sleep, stages 3 and 4 are the ones that you should be trying to get to as much as possible. If you ever go to sleep and then you wake up and you don't feel rested, it's because you actually haven't gotten to slow wave sleep or stages 3 and 4. And again, you tend to go through these stages in another type of cycle, which is known as an ultradian rhythm. And these ultradian rhythms are small rhythms. And these small rhythms last like 90 minutes to maybe an hour to 90 minutes at a time. And these small cycles are what I'm going to be showing you next. And again, even this last point here, to be honest, there's a lot of debate as to whether or not REM sleep helps you to remember things better or not, and it's still very, very controversial in the field. So this is sort of a typical cycle, and you can see that these cycles occur at about 90-minute intervals. And there's a couple of things here. You don't have to memorize it. I'm hoping most of it is somewhat of a review if you've had it in first year psychology or even back in high school. I'm sure you talked about sleep. So don't bother memorizing this. I'm hoping most of it is a review for you. So if you're taking a look at a typical individual who goes from an awake state and then they start activating the VLPO group of neurons, they'll go into different levels of sleep. So they might go through REM, NREM sleep, or stage 1, stage 2, then go all the way down to stage 4. It doesn't last very long. You're not in this period of sleep for very long, and then you come out of this period of sleep, and then you go through this entire cycle again. And you're in these different levels. A typical individual will go through 4 of these cycles during the course of an evening. I'm not so sure it's relevant for your generation anymore, to be quite honest. I think most of you are still awake at 12 o'clock at night, and I don't think that you're actually up at 7 o'clock in the morning. And my guess is that this whole cycle is a lot different. But a normal individual will get about 4 of these different cycles. And one of the things that you will note from this is that as you go through these cycles, in this NREM period of sleep, as you get closer and closer to when you need to wake up, you actually spend more and more time in NREM sleep. The other thing that you don't do is you don't go into deeper type 3 and type 4 stages of sleep. And so by the time you're waking up, actually you are not in those deeper stages of sleep. And you start off in these light stages of sleep, you cycle through, but by the time you wake up, you're no longer in those deep stage 3 and stage 4. So again, nothing for you to really take away from this particular slide. A couple of things, again, I just mentioned in passing. The ultradian rhythms are not part of the suprachiasmatic nucleus. There is a faster oscillator that is involved. So it's not a 24-hour oscillation. It is a 90-minute oscillation. It's believed to be influenced through the reticular activating system. And again, it's the suprachiasmatic nucleus that actually tells you that you should be going to sleep when it is dark out versus when it is lighter out, and it starts this whole process off. I am going to tell you the next slide here is actually one that I'm going to ask you to make sure that you know, and this gets into this idea of sleep atonia. Most of you will have seen sleep atonia before. And this is a very interesting phenomenon, and it goes back to what we know about motor neurons, what you know about hyperpolarization, etc. So reticulospinal, is that an ascending or descending pathway? So it's going from the reticular formation down into the spinal cord. How about corticospinal? You guys are going to do really well on Tuesday, right? I know. Like you're just playing with me right now. I know you guys know. So this is a motor control pathway. It involves controlling your muscles. And these are very active in REM sleep that I just went through. And if you go through these different cycles, you'll note that we don't spend a lot of time in this REM sleep but we do more and more as we get closer to actually waking up. But during the time when your eyes are moving really rapidly during REM, where we think you might be dreaming or you might be consolidating memories, during this period of time, it's just eye movement. Your body actually doesn't move very much. So when you're in REM sleep, your body has what's known as sleep atonia. I could pick up your arm and then it would just fall down. And again, this is surprising because these reticulospinal and corticospinal tracts, which we know are involved in motor control and are involved in muscle contractions, are very, very active at this period of time. So why isn't your body moving around all over the place? Like generally speaking, when you're in REM, when you might be dreaming or might not, again, theory is very controversial, why aren't you like thrashing around? It happens, but it's very, very rare. One of the reasons why we think that this sleep atonia, where your body doesn't have muscle tone associated with it, the motor neurons that are in your spinal cord are all hyperpolarized by inhibitory reticulospinal groups of neurons, the pontine neurons that are found in the pons. So even though you have descending signals that say that your muscles should be contracting, another group of reticulospinal neurons from the pontine region, the pons, is actually hyperpolarizing all of your spinal cord neurons. It makes it harder for them to be excited. It makes it harder for them to be active and to contract your muscles. And this is what we think accounts for sleep atonia. And again, 10 millivolts may not seem like a lot, but it's enough to inhibit activity of the muscles that would normally be activated by these spinal cord motor neurons. What makes this incredibly interesting is this phenomenon where you will be in REM sleep, where in REM sleep, the reticulospinal neurons from the pons are actually inhibiting your spinal cord motor neurons, and you've woken up from REM sleep. And have you ever had that phenomenon where you've woken up but you can't actually move your muscles? And this phenomenon is actually, for some individuals, really, really scary because it's like sleep paralysis, right? You wake up and you can't do anything. Imagine how scary that feeling might be, especially in the middle of the night. So this sleep atonia, where the spinal cord motor neurons are inhibited by a particular group of spinal or reticulospinal neurons from the pontine nucleus, are really important. And these inhibitory pontine cells work and cause this sleep atonia, and in addition to that, will also activate a number of other systems or inhibit a number of other systems. So these two systems that I'd like you to make sure that you know that cause this sleep atonia. A couple of other modulatory systems that we've looked at before. The cholinergic system. Do you guys remember where it originates? What group of neurons in the brainstem does it originate from? Remember we were going to work on that short-term memory? It seems our experiment didn't work well. Any ideas? Can I suggest very strongly by next Tuesday that you learn all of these brainstem nuclei that project and cause the release of these different neurotransmitters? Within the midbrain, this group of cholinergic neurons actually supports waking. It makes sense because cholinergic neurons help you pay more attention, are involved in things like vigilance, etc., along with the adrenergic pathways. During the time that you're actually going to sleep, where these pontine inhibitory cells are becoming active, they will also start turning off the cholinergic fibers. So the cholinergic fibers, as you're drifting off to sleep, become less and less active. And again, this is through those same groups of these inhibitory pontine nuclei. The serotonergic as well as the noradrenergic systems, they are most active when you are awake. And as you start going into REM sleep, and as you start going into the stage 1 through 4, it's believed that these pontine inhibitory neurons are also going to be acting on these modulatory systems to turn off the serotonergic and adrenergic systems as well. So sleep is actually a really complex phenomenon. It involves neurons in the suprachiasmatic nucleus. It involves things like the TMN and activation and the inhibition of TMN neurons when you're sleeping, their activation when you're starting to wake up, a number of hyperpolarizing events in your muscles, things like reduction in cholinergic activity, serotonergic activity, etc. So I'd like you to make sure that you're aware of all of those different things. Again, one of the things I'm going to recommend to you, please don't leave this to the last minute, because the term test that's coming up, there's a lot of material. I'm not trying to make it purposely harder than the first term test was. It's going to be more or less the same. And as long as you've gone over your slides, you'll be okay, but you need to go over your slides. Please start as soon as you possibly can.